Welcome at Medical Sciences, made easy by Naphtali Muhumuza. And in today's session, we want to talk about urea cycle. Urea cycle is under protein catabolism. We normally see that these proteins, they are majorly catabolized in the liver by two processes, major two processes, that is transamination, whereby we are transferring the amino group from alpha ketoglutarate, forming glutamate and pyruvate. And these transamination reactions, they are catalyzed by ALT and AST, what we call alanine aminotransferase and aspartate aminotransferase. We are able to form glutamate, and this glutamate can undergo deamination. So we are saying that the product of deamination, with the product of transamination reaction, we can use them under deamination to form ammonia. And ammonia is always the precursor of urea cycle. What is urea cycle? Here it is whereby we are converting toxic ammonia to non-toxic urea so that we can easily excrete it via urine. So this one is majorly looking at how we convert the toxic ammonia formed by either deamination or non-oxidative ways to form ammonium, and this ammonium is detoxified under urea cycle, which occurs principally in the liver. So we see urea cycle occurring in the liver. So this cycle occurs in the liver. Cytosol, both the mitochondria and the cytosol of the liver cells. So we see in tissues like brain, like the kidney and the liver, doing the process of deamination. What is deamination? Here, deamination is whereby the L-glutamate amino acid is being oxidized to alpha ketoglutarate, producing ammonia, whereby enzyme known as glutamate dehydrogenase is catalyzing this oxidation by removing hydrogen group from glutamate, forming alpha ketoglutarate and ammonia. And we said that this deamination process is producing ammonium compound, which is very toxic to our body. So how does the body deal? For example, if this ammonia is formed in the kidney or in the brain, how is it transported safely to the liver? Because we have seen urea cycle occurs only in the liver cells. So for safe transportation of this ammonia formed, it is transported in the form of glutamine. Whereby if I have glutamate, combines with ammonium ions to form glutamine. And this reaction occurs in the presence of glutamine synthetase. And this glutamine is the one that is transported to the liver. It's the one that is taken to the liver cells. And once it reaches in the liver, this glutamine, which is formed, the glutamine which is formed in the liver, it is converted back to ammonium ions plus glutamate. Because in the liver we see an enzyme, and this enzyme is known as glutaminase. Glutaminase catalyzes the conversion of glutamine to ammonium ions and glutamate in the presence of water. So this is how we can transport the ammonia formed in the kidney and the brain, how we can transfer it to the liver, whereby it is first converted to glutamine, and when glutamine reaches in the liver, it is converted back to ammonium ions and glutamate by glutaminase enzyme in the presence of water, so that these ammonium ions are transported safely to the liver cells. And what happens to ammonium when it reaches in the liver? This is what we are calling urea cycle. That in the liver, these ammonium ions produced from deamination, they are able to cross the mitochondrial and enter into the mitochondria. So we see the ammonium ions together with the carbon dioxide in the liver cells. All of them, we see them entering, whereby this ammonium enters and combines with the carbon dioxide. And after combining with the carbon dioxide, we form a compound known as carbamoyl phosphate. Carbamyl phosphate. And how does combination of ammonium ions and carbon dioxide combine together to form carbamoyl phosphate? We see there is addition of a phosphate group. And this phosphate group is coming from hydrolysis of ATP, whereby we are utilizing 2 ATP molecules to form AMP plus to form ADP plus phosphate inorganic. This one occurs in the presence of magnesium as a cofactor, and the enzyme that catalyzes the joining of ammonium ions and carbon dioxide, this enzyme is known as carbamoyl phosphate synthase type 1. Carbamoyl phosphate synthase type 1, because type 1 is the one that is found in the mitochondria. Type 2 is found in the cytosol. So carbamoyl phosphate synthase type 1 is the one that catalyzes the combination of ammonia and carbon dioxide to form carbamoyl phosphate, whereby it involves hydrolysis of 2 molecules of ATP and magnesium ion as a cofactor, forming carbamoyl phosphate. And this reaction is the first reaction in urea cycle. And after forming carbamoyl phosphate, the second step is conversion of this carbamoyl phosphate to citrine. We are going to convert carbamoyl phosphate to citrine. And how do we do it? Is that the end product of urea, we always see ornithine. And this ornithine enters into the mitochondrial matrix and combines with the carbamoyl phosphate to form citrine. And the enzyme that catalyzes this reaction is known as ornithine transcarbamylase. Ornithine transcarbamylase is the one that catalyzes the transfer of ornithine group to carbamoyl phosphate to form citrine. This is the second step in urea cycle occurring in the mitochondria. Ornithine transcarbamylase enzyme catalyzes the transfer of ornithine, which is the last product of urea cycle, to carbamoyl phosphate, forming citrine. And the enzyme, we are calling it ornithine transcarbamylase. And this ornithine transcarbamylase, after forming citrine, this citrine can move outside the mitochondria. It exits the mitochondria, and we find it in the cytosol. And this citrine is going to form adenosicinate, and this adenosicinate formed is going to be converted. This adenosicinate, we are having citrine forming adenosicinate, and from adenosicinate, we are going to cleave it to form arginine, and after cleaving this adenosicinate to arginine with the release of fumarate, which is taken to the Krebs cycle, before we convert this arginine to ornithine with the release of urea. And these steps occur in the mitochondria. So we are going to see that citrine, which when it reaches in the cytosol of the liver cells, we combine the second amine group. There is the addition of the second amino group. And this amino group is derived from aspartate amino acid. Aspartate amino acid donates the second amine group. The first amine group is donated by glutamate Then the second is denoted by aspartate, whereby this amine group we added to citrulline in the presence of an enzyme known as agenosuccinate synthetase, or we can call it synthase, whereby agenosuccinate synthase catalyzes or adds the amine group, then involving hydrolysis of ATP, whereby ATP is being hydrolyzed to form AMP plus the pyrophosphate. And this one also needs magnesium as a cofactor, whereby this agenosuccinate synthase adds the second amine group to citrulline, presence of ATP, to form agenosuccinate in the cytosol. So this is the third reaction. This is the third reaction of the urea cycle. And after forming agenosuccinate, this agenosuccinate formed undergoes cleavage. It is cleaved. So second point number 4, we are going to see cleavage, whereby we are going to cleave agenosuccinate to form arginine and fumarate. And the enzyme that catalyzes this reaction is known as agenosuccinate lyase. Agenosuccinate lyase, or you can call it agenosuccinase. So agenosuccinate lyase enzyme catalyzes the cleavage of agenosuccinate to form arginine and fumarate. And this fumarate is an intermediate in Krebs cycle, or what we call TCA cycle. It can be used in Krebs cycle to provide energy, or intermediate. Then the arginine, which is formed from the cleavage by agenosuccinate lyase, we form arginine, and this arginine undergoes a condensation reaction whereby we add water. It is addition of water in the presence of an enzyme known as arginase enzyme, whereby this arginase enzyme will cleave off the ornithine and the urea. And our major interest is urea, and its molecular formula of urea is this, whereby we have the two amine groups, one amine here, then this is another amine group. The first one from glutamate, the second one from aspartate, and we are able to form our urea. So this is the urea formation. How do we do it? We have the first two steps in the mitochondria, whereby we had ammonia from deamination being taken to the mitochondria, combining with the carbon dioxide to form carbomyrophosphate in presence of carbomyrophosphate synthetase type 1, or synthase type 1. Then this one, in the presence of ornithine transcarbamylase, it is transferring the ornithine to the carbomyrophosphate to form citrulline, which exits the mitochondria and goes into the cytosol. And the cytosol, we add a second amino acid from aspartate in the presence of ATP and agenosuccinate synthase to form agenosuccinate. And agenosuccinate in the cytosol undergoes cleavage by agenosuccinate lyase enzyme to form fumarate and arginine. Fumarate goes into the Krebs cycle for provision of energy. Then arginine undergoes condensation reaction, or whereby we add water to form ornithine and urea. Ornithine continues to form more citrulline, then urea is cleaved off and it is taken to the liver, where it is excreted. It is taken to the kidney, rather. It is taken to the kidney and it is excreted via kidney. As we know that urea is being smaller in molecular size, it is both secreted and it is both and reabsorbed. So that's how we excrete this ammonia, which is toxic, to non-toxic urea by the process known as urea cycle. So this is so in summary, we have how many equations? If I can remove this part, we have seen there are majorly few equations, whereby the first one is that ammonium ions react with carbon dioxide to form carbamoyl phosphate in the presence of carbamoyl phosphate synthase type 1, which occurs in the mitochondria with hydrolysis of 2 ATPs and magnesium as a cofactor, forming ADP plus phosphate in organic. The second reaction, we have seen carbamoyl phosphate combining with ornithine, combines with ornithine to form citrine. This citrine, it occurs in the presence of an enzyme known as ornithine transcarbamylase. Then the citrine, which is formed step number 3, and these first 2 steps occur in the mitochondria of the liver cells. Then these ones, the cytosolic reactions, we are seeing the citrine formed in the cytosol. It combines with another ammonium from aspartate, forming agenosuccinate. In presence of agenosuccinate synthase, where we have seen also hydrolysis of ATP to form AMP plus a pyrophosphate, presence of magnesium ion as a cofactor. Then the 4th step, we have seen agenosuccinate being cleaved to form arginine with removal of fumarate, which enters Krebs cycle. Then we have seen number 5 is whereby arginine, which is formed in the presence of arginase enzyme with addition of water. This arginine is cleaved. Now this one was in presence of agenosuccinate lyase, which was cleaving off the fumarate. Then arginine in the presence of water and arginase enzyme, we are able to form ornithine, and we cleave off the urea. And this is the one which we are interested in. Then the ornithine which is formed goes back to combine with the carbamoyl phosphate to form another citrine. So in summary, this is the summary of both urea or urea cycle. You can summarize these into steps, whereby in these steps, 5 of them, whereby the first 2 occur in the mitochondria, whereas the last 3 occurs in the cytosol. And whenever you are born with genetic disorders, whereby you are lacking carbamoyl phosphate synthase type 1, you can suffer from what we call hyperammonemia type 1. So deficiency of this enzyme leads to hyperammonemia type 1, whereby ammonia increases in blood circulation, leading to Alzheimer's disease. Because we said ammonia is very toxic. It can even destroy the brain. It can cross the brain barrier and causes Alzheimer's. Alzheimer's is whereby it is characterized by forgetting or is not paying attention. Then when you are lacking ornithine transcarbamylase, that is hyperammonemia type 2. Others have citrinemia when they are lacking synthase. We can even see others having agenemia when they are lacking agenase. So this is deficiency leads to agenemia. This one leads to citrinemia. This one is hyperammonemia type 2, and this is hyperammonemia type 1. Thank you so much for listening. This is Medical Sciences Made Easy by Naphtali Muhumuza. Always remember to subscribe and always watch until the end. Thank you so much.